Hello friends, welcome back to my channel Biofrig. In today's lecture video, we will be talking about starch and sucrose synthesis. So we basically saw what is light reaction, then the products of the light reaction were used into the C3 cycle or the Calvin cycle, and in the end of the Calvin cycle, we got something called as glyceraldehyde 3-phosphate, and then we also regenerated RUBP. But now here we are interested in this glyceraldehyde 3-phosphates, which are nothing else but triose sugars. Okay, now we are learning that plants prepare their own food since 7th standard, and that's photosynthesis, isn't it? But this is it, this is the food? No, there is a separate procedure wherein the starch is synthesized, the sucrose is synthesized, which is the proper form of food, the energy molecule in the plant. And the sucrose that I'm talking about is important for the transport. Okay, it's easy for the transport. Although both are the form of food or the energy molecules there, sucrose is easy for transportation and it is synthesized in the cytosol. Okay, while the starch that I'm talking about, it is stored by the plant in vacuoles or something like that, but it is important storage molecule, energy storage, just like how we store our food for like months and for a year we plant something, right? Similarly, when plants have some storage there wherein they'll store the starch, and starch that is there is synthesized in the chloroplast. So to remember that starch has seen it, so it is synthesized in chloroplast. That's how you can remember. And other thing is sorted later. So the sucrose that is there, it is synthesized in the cytosol. So let's get started from this triose sugar that I'm talking about, nothing else but glyceraldehyde 3-phosphate. Okay, let's form first starch in the chloroplast. However, before starting to that, I want you guys to understand that in the reduction phase, we had got 6 molecules from the 6 molecules of glyceraldehyde 3-phosphate from the 3 molecules of RUBP. Out of those 6 molecules, I told you that 1 molecule will be used in this process, that is synthesis of sucrose or starch, and other 5 molecules went into the regeneration phase. So this is the step wherein the things bifurcated into the 2 directions. So now let's get started with the glyceraldehyde 3-phosphate. Now let's take here 2 molecules of glyceraldehyde 3-phosphate. These 2 molecules of glyceraldehyde 3-phosphate, which are 3 carbon each, will form something called as fructose 1,6 bisphosphate. Okay, fructose 1,6 bisphosphate will be formed from this 2 molecules of 3 carbon compound glyceraldehyde 3-phosphate by the enzyme aldolase. So from this fructose, the next compound that is formed here is fructose 6-phosphate. I'll just write the short forms. Okay, so how this is formed? Basically, one phosphate has to be released, inorganic phosphate, right? So that is done and you get fructose 6-phosphate. So from this fructose 6-phosphate, we form something called as glucose 6-phosphate. So here what are we doing is these 2 are isomers of each other. So which enzyme will come here? Hexose, both are 6 carbons, so hexose isomerase. Okay, so from glucose 6-phosphate we obtain something called as glucose 1-phosphate. Now how do we get glucose 1-phosphate? We are just shifting that 1-phosphate group from carbon 6 to carbon 1. So this is phosphomutase here. Okay, so from glucose 1-phosphate there comes a turning point. Now this is the step which is important. From the step, what we will get is ADP glucose. Since we are getting ADP glucose and there is no phosphate, it's obvious that we don't want this phosphate group there. So here, another thing that I can observe is I have ADP there. So for that matter, what I'll do is that I'll break one ATP. So from this ATP, what I'll do, I'll remove one phosphate now. I'll remove one phosphate, and this phosphate this time is not utilized. Remember that this one phosphate and one phosphate from here will be removed in the form of pyrophosphate. This pyrophosphate that I have can be later converted into the orthophosphate. Two orthophosphates will be released by pyrophosphatases enzymes. Okay, so this is something called as orthophosphate. Okay, this is just in addition to that, what happens to that pyrophosphate, I've given that. Now this ADP glucose that I have, that can be easily converted to our final molecule there, that is starch. So this can be stored later. Now this is done by starch synthase enzyme. Okay, which is the enzyme that's acting here? Starch synthase. Okay, so this is basically the starch synthesis occurring in the chloroplast. So what is the first thing happening here? Glyceraldehyde 3-phosphate to fructose 1,6 bisphosphate, 1,6 bisphosphate to fructose 6-phosphate, then to glucose 6-phosphate, then glucose 1-phosphate. That phosphate has to be removed and another phosphate from the ATP will be removed to get your ADP glucose. From ADP glucose you can form starch. Now let's talk about the sucrose synthesis. Now in the beginning of the sucrose synthesis, there has to be some sort of transporter here. Through this transporter, your glyceraldehyde 3-phosphate here, which was there, okay, this glyceraldehyde 3-phosphate which is getting formed in the chloroplast will move out. Okay, that is your triose sugar there. Now this triose sugar will follow the same path till here. That is, glyceraldehyde 3-phosphate will get converted to fructose 1,6 bisphosphate, then it will get converted to fructose 6-phosphate, then it will get converted to glucose 6-phosphate, then it will get converted to glucose 1-phosphate. Now from here the things change. From here what happens is that we use something called as UTP, not ATP. We are using UTP, and pyrophosphate will be released. This phosphate will be removed. Phosphate from UTP will be removed, and what we will get is UDP glucose. Okay, we got UDP glucose. Why U here and why A there in the chloroplast? Because this is in the cytosol, and if you remember that there'll be uracil there, UTPs will be prominent there, right? Now from this UDP glucose we can convert this UDP glucose to sucrose 6-phosphate, and from the sucrose 6-phosphate we can form sucrose. So this is just the extra thing that you should remember, and that UTP part. Now UDP glucose is converted to sucrose 6-phosphate, okay, by sucrose 6-phosphate synthase enzyme. Okay, synthase doesn't really use ATP there, while synthetases they use ATP. This is just extra info for you. Now here the conversion is carried out by sucrose 6-phosphate phosphatase. Okay, so here phosphatase will be used because we are removing the phosphate group. Another orthophosphate will be removed. So this is the overall process of starch and sucrose synthesis. I hope you found this lecture helpful. I made it I tried to make it a bit simple, because till here if you go to see, all these steps are very much similar to the starch synthesis, and once you get used to it, once you practice this, you will remember the names as well. And just remember there is UTP or ATP will be used in the chloroplast, and just this extra steps, and you will be done with the starch and sucrose synthesis. So guys, see you in the next lecture. Till then, just keep learning, keep revising, and see you soon.